Hello and welcome to Freedom from Fear, a German New Medicine podcast. I'm your host, Ashley Newberry.
And I'm your other host, Abigail Puccioni. Please be aware that nothing discussed here on this podcast should be taken as medical advice.
Consult your trusted professionals and your own inner knowing before making any health decisions.
Hey guys, so before we dive into the episode, we just wanted to share some really exciting news about an event that we're going to be a part of this January 25th through 26th in Austin, Texas, and it's called Paradigm Shift, a GNM Weekend, and this is truly going to be all about building GNM community and socializing with like-minded individuals who are all on this paradigm shift journey, and we're going to feature a lineup of expert speakers on a wide variety of different GNM topics. Ashley and I are both going to be speaking at this event. And the main focus is going to be on how to actually integrate this beautiful body of knowledge into your life. We would really love to see you guys there, and we have the website linked up in the podcast description. It's paradigmshiftweekend.com. So definitely check that out for all the details, including the full speaker lineup and information on how to register. So yeah, we hope to see you guys in Austin this January. Hey everyone, welcome to today's episode where we have a very special guest that we're excited to introduce to you. So this special guest, Mark Gober, is actually going to be speaking at our upcoming Paradigm Shift event in Austin, Texas. So Ashley, Mark, and I, and a bunch of other speakers are all going to be there. So make sure and check out the tickets for that if you're interested in coming and attending in person. That's going to be a great event. So anyway, for today's episode, this is a very, very highly requested topic, all about contagion and germ theory and questioning what we have been told to believe about the seeming phenomena of contagion. So we're going to be unpacking this topic in depth, and I think you guys are really going to like it. So Ashley, do you want to introduce Mark?
Yes, thank you. So today we're excited to welcome Mark Gober, author of several award-winning books, including An End to Upside Down Medicine, which we'll be exploring in today's episode. He previously hosted the podcast Where's My Mind and serves on the board of Institute of Noetic Sciences. He graduated magna cum laude from Princeton University and has worked in Silicon Valley's tech industry and as an investment banking analyst in New York. Welcome, Mark.
Thank you so much for having me.
Yay. So let's just jump right in. You can talk a little bit about your book, An End to Upside Down Medicine. Ashley and I have both read that. Ashley and I are also going to be asking questions throughout this episode and then the next episode, because keep in mind, guys, this is only part one. So in this episode, we're going to be unpacking basically germ theory and talking about, is there actual scientific evidence to prove that viruses even exist? So we are going to be talking about that in today's episode. And then next week, we're going to have an episode come out, part 2, that's going to be a continuation of this conversation, all about, okay, so what is the cause of the seeming phenomena of contagion? If it's not germs, then what is it? So just a little sneak peek there. Stay tuned. But yeah, I also wanted to mention that in all of Mark's books, he's written several books. And Mark, I believe you've mentioned German New Medicine in all of them. Is that correct? Or almost all of them?
Just the two most recent books. So An
Two
End
most
to
recent.
Upside Down Medicine, where I discuss GNM a bit, and I mention it in my latest book, An End to the Upside Down Cosmos. I actually was not aware of GNM until I started researching for the medicine book. So I'm
Okay.
newer to this subject.
Okay, awesome. So we're really excited to hear later on in this episode and the next episode where German New Medicine, fits into all of this and your own research and journey. So yeah, let's just dive into your book and talking about germ theory and how we've been raised to believe as far as understanding viruses and contagion.
Yeah, let me take you back through the history of how I even came to approach this topic, because I had heard people questioning germ theory since about 2020. And that's when everything started with COVID. And there were a range of people who were questioning the narrative at that time. Some people said COVID is not as severe as people believe, but it is a virus and you can get sick and maybe some people will die. But even that was controversial to say back in 2020, to say, look, it's not that bad. It's sort of like the flu. And then there were other people who said, this is really an agenda to get people to take a vaccine. Yeah, there's a virus. It's not that bad, but this is a vaccine agenda. And that was super controversial back then. And then there were a few people who said, yeah, it's true. This is not that severe, and there's a vaccine agenda, but also there is no such thing as SARS-CoV-2 that is causing the illness called COVID-19. And I remember hearing that and thinking, well, that's quite the claim, given that the whole world is being shut down on this basis.
Yeah.
But the arguments that were being made, and I initially heard it from Dr. Andrew Kaufman, who ended up being one of the reviewers of my book. So he's a traditionally trained medical doctor. And then Dr. Tom Cowan, and also Doctors Sam and Mark Bailey in New Zealand. So there are traditionally, classically trained doctors who have been speaking about this topic. But when I heard the arguments back in 2020, my reaction was, I can't really debunk what they're saying because they're speaking in technical terminology about methods used in laboratories. But it seems really far-fetched. And what some people claimed is that it was a psyop, a psychological operation to steer the freedom community away from the people who are legitimately questioning vaccines, and it would make them sound crazy. So they would lose legitimacy for the things that actually mattered. So I had this seed in the back of my mind. But prior to that, and I'd written 2 books previously, my first one, An End to Upside Down Thinking, then An End to Upside Down Living. And I had heard about alternative notions of health, which we'll talk about in the next episode more, the notion that consciousness affects our health and energetic type energy healing, those sorts of things. So I was definitely thinking about health, but I'd never heard about a question to germ theory. And that's really what this was about. But in particular, I was just hearing about the notion of a virus, that yes, they were saying that SARS-CoV-2 doesn't exist, but they even expanded it further and said, actually, there's no evidence that any virus exists at all. So I was like, whoa, that's kind of crazy. And so I kept this in the back of my mind. I was writing a bunch of other books. And then I went to a conference in Tennessee in 2023, and Alex Zeck was there. He hosts the podcast, The Way Forward. And he was basically saying, Mark, you've got to look into this germ theory thing. He gave a talk on virology, showed how no virus has ever been established. So I was thinking to myself, we're a few years into this and people are still talking about it. And it's not like it's not losing momentum. It's actually gaining momentum somehow. And so that sparked something in me to say, okay, like, why would that be the case? If this were easily debunkable, wouldn't people be leaving it? And then Alec and Mike Winner, they asked me to be on their series called The End of COVID. It's still available at theendofcovid.com. It's basically a library of maybe like over 100 hours of content on germ theory, but also the topics that I was asked to speak about were more in my political areas. So the notions of liberty and the Great Reset. So I was asked to speak on those things. I said yes. And then when it came out in the summer of 2023, I listened to the other talks, and it included these talks on germ theory and whether viruses have been established. And the more I looked at it, the more I said, wow, there's something really big here. And I was super resistant to it. I'm like, no, this can't be true. Like, is it true that germs actually don't cause disease? Let me take a step back. Germ theory is the idea that microbes, these microscopic organisms, whether it's a virus or a bacterium or something, can cause disease in a healthy organism. But another part of that that's being challenged is they would say bacteria exist, but they don't cause disease. Those who question germ theory. Bacteria do exist, they don't cause disease. Viruses don't even exist. And they might say that parasites and fungi, they exist, but they don't cause disease either. That's the challenge to germ theory. And I was hearing this stuff like, okay, this is really big, because it does relate to my interest in health and consciousness, because I have been interested in like what actually makes people healthy and sick. And the more I looked at it, the more I said, this is a really big issue. And I said, okay, what I'm going to try to do is convince myself of the arguments I'm hearing by writing the book. Maybe I won't publish it. Maybe I will. I don't know if I can actually publish it, because people say that the no virus, as it's called, community is just a psyop, like I said before, and I don't want to be associated with the psyop. So I was going through all this stuff in my mind, and I'm giving this preface because it didn't come easy for me to get to where we're going to get to in this conversation. But I got to the point where I thought the logic was very strong. And I came to the general conclusion, which we'll dive into, that a virus, which is believed to be a replication competent intracellular parasite, meaning something that gets inside the cell, replicates, and causes disease, and then goes from one person to another, that entity has actually never been established using the scientific method, meaning there are no studies establishing the existence of this thing where there's an independent variable and proper controls. So
Mhm.
that's point one. And then point two is that the microbes that have been established, like bacteria, there is no scientific evidence that they cause disease in healthy organisms. They do exist and they can show up in large quantities, but the question is whether they are actually causing disease or whether they are showing up in large quantities in the body because they're acting as a cleanup crew, or they are in some way helping the body rid itself of some kind of toxicity, and therefore they're needed in large quantities, in which case we would be blaming the wrong root cause by blaming the bacterium or whatever other microbe is there. So this
Mhm.
is some would call this alternative view the terrain model, that it's not that our health is governed by the terrain of the body, in the same way that if we want to grow plants, we want the soil to be really well taken care of. The terrain idea is that we want to take care of the body and also the mind. I would say the mind is part of the terrain. So it's this physical, psycho-spiritual entity, and if you take care of that, then the body's going to be okay. And there's a
Mhm.
hybrid in the middle that I should mention, because some people will say, yeah, the terrain model makes good sense, and it will help you better fight bacteria and viruses if your terrain's in better shape. That's not terrain theory. That's a hybrid. It's really still based on the notion that microbes can cause disease and that viruses even exist. So I'll pause there.
Yeah, yeah, you made some really good points. And um, I can't remember exactly what you said in the book, but when you were describing the virus and the intracellular um, uh, thing, that's inside the cell that we're calling a virus, that you questioned, well, if we're only seeing it inside of the host cell, how does it ever get to the next cell? Like how does it, if we haven't been able to isolate it, how is it that it travels from one cell to the next, like in the in-between time? So I'd love to hear if you have any more research on that part, because I think for our listeners and people that are newer to exploring this topic, right? It's quite the paradigm shift
Yeah.
to see it that way.
Yeah, so there are some knee-jerk reactions that people will push back with, and we should get into the notion of isolation, which is a bigger topic too. But some people will say, well, of course you wouldn't be able to isolate a virus by itself because it's a parasite that's inside of a host cell. So it's always going to be in the host cell, which is a convenient counter argument because you could never disprove that. The problem with that argument is part of what a virus is supposed to be is something that gets inside of the host cell and then presumably leaves the host cell as it's spreading.
Right.
So wouldn't you be able to capture the virus before or after it's a parasite inside the cell? And then another counter argument sometimes people will make is, yeah, it's really hard to isolate a virus because it's so small, or maybe there's not enough of it there. And yet at the same time, it's supposed to be so contagious that if you're in the same room as someone, you might get some of it in your body, and it can cause this disease that can kill you. So there's this paradox that always seems to come up in those who defend the virus paradigm versus what we actually experience in reality. There's this mismatch. And that was powerful to me because I was looking for the counter arguments of, let's just see a
Mm,
mainstream virologist debunk all this stuff. Do it
yeah.
quickly. And I never got a good answer. And I'm still looking for it. And the answers I get are, well, we just have to use indirect evidence. That's the best we can do. And as we'll discuss today, that there are big problems with that mentality.
In your book, you listed a lot of different examples where it seems that those in the conventional, let's say, I don't know, science or medical communities that are the proponents of this germ theory. And yes, of course, viruses exist. And if you believe that they don't exist, or if you even question it, you're insane, basically, and you're anti-science or whatever. Yet they seem to be super evasive. Like they can't provide the evidence. I know that you
Mhm.
mentioned, I forget exactly which doctor it was, but I think there were several that you mentioned in your books that made these, what are they called? Like requests for information? What, ROIs? Yeah, where,
Freedom of
yeah.
information requests all over the world.
Oh, okay. Yeah,
Yeah,
that's what I'm talking about.
that was really powerful to me. I think that was actually one of the turning points in my research when I saw that and this is run by a woman named Christine Massey in Canada, who's working with colleagues all over the world to submit formal requests to the CDC in the US, all the top health organizations, to say, can you show me a purified sample of just a virus?
Mm-hmm.
Not putting it through a cell culture medium and then making a bunch of inferences. Show me a purified sample. And all over the world, every organization is saying we don't have any records for that. In fact, one example that I quote is what you're describing violates the gold standard of how we do virology studies, and therefore there's no way you're going to get an answer to this. I'm paraphrasing. And this really gets to the core of the issue, which is that what's known as the gold standard since 1954 is a method that does not necessarily show that there is a virus. The results of the gold standard studies could be explained by non-viral means. So you end up with a house of cards, basically, where assumptions are made based on the way certain studies are conducted. And many doctors I've even spoken to, they're not aware of how these studies are conducted. They're not aware of the fundamental 1954 paper or even what led to it. And so we end up with making assumption after assumption based on the belief that this virus exists. And there's a logical fallacy. It's known as the reification fallacy, where you speak of something as if it's real. And by continuing to use the word, you're basically reifying its existence.
Mm-hmm.
It's almost like saying, like, instead of virus, we could say flying unicorn or some other term that hasn't been established. But because people use the term virus over and over again, it's almost like it reifies the fact that it's real. But if we look at the way the studies are done, it hasn't been established using the scientific method.
Yeah. Yeah. And I just wanted to make a quick note. I think you mentioned Louis Pasteur in your book, if I remember correctly. And even, you know, the studies or what he was researching, which I don't remember what year that was, but a long time ago, you probably remember. You know, like it was like his best guess. And then all of this stuff is based on someone's best guess that's not even right, that doesn't even fall under the scientific method. And yeah, we just keep reaffirming this thing that was kind of based on not science, right? And it's wild.
Yeah, the history here is really important. And I go through this in the book and into Upside Down Medicine. Like, how do we get to this point of believing in this intracellular parasite? Because certainly long ago, the technology did not exist to see something like that. And yet the term virus existed. It meant something different. It meant a slimy poison. And in the book, I give different dictionary definitions of what virus meant. But when Louis Pasteur was working with rabies in the late 1800s, he saw symptoms. So a very important point here before we continue is that we're not disputing whether people get sick or whether they die from symptoms. The question is what's causing that? Is it a virus or something else? I know that can trip people up. So we're not denying illness. So and then Louis Pasteur saw illness and he couldn't find a transmissible particle that was going from one organism to the other, but he said it must be there. There must be something going from organism to cause these symptoms. He didn't have the technology at the time to see something as small as a virus is now believed to be. And a virus is believed to be much smaller than bacteria are. And it wasn't until the 1930s, and then this technology became more prevalent in the following decades, the electron microscope was invented to be able to see things as small as a virus is alleged to be. So anything before that is a guess. If they're talking about a transmissible agent as small as a virus, that's important to keep in mind. But then around the 1950s, things shifted for multiple reasons. One was this technology, the electron microscope, to be able to see things much smaller, but also the 1953 double helix structure of DNA paper by Watson and Crick, because that then gave the impetus for creating an organism that's DNA based that could replicate. And this was a point made by Stefan Lanka, who was a classically trained virologist who is one of the leading, most outspoken people saying that no virus has ever been established. And that was powerful for me to say, wow, this guy was classically trained, and he's saying we've never established this. But he talks about the Watson and Crick paper being very fundamental, 1953. So all of a sudden we've got DNA, something that could replicate. Now, if we're trying to understand disease, that was the goal, why are people getting sick? Well, maybe it has to do with DNA. And then in 1954, this was the foundational paper, Enders and Peebles. This is the gold standard method for quote unquote virus isolation. And really what they've done is redefined isolation to fit virology, because they're not talking about isolation using the traditional definition, which means to separate one thing from other things. And to quote Dr. Tom Cowan, basically, he says, if we want to isolate a hammer from a toolbox, we take the hammer out of the toolbox and then we can characterize the hammer. We can do all sorts of studies and understand what the hammer is doing. It sounds simple, but you'd expect that that's what they're doing with virology studies. That's not what they do, however. When they say isolation, they're referring to the method done in this 1954 paper. And by the way, Enders won a Nobel Prize for something else later that year. So this was very prominent stuff. And they were looking at the measles alleged virus. And the methodology is basically something like this. And it might vary slightly depending on the studies done, but this is how virus, quote unquote, isolation is done all over the place now. The researchers take fluids from a person who's sick, and sometimes they might filter those fluids, but they're fluids from the person who has symptoms. And then they put those fluids into a soup, which is known as a cell culture. And that cell culture has a bunch of stuff in it before the fluids from a sick person's put in. It has antibiotics, it has monkey kidney cells, and all sorts of other things. And what the researchers do is they want to see if cells break down in this soup when the fluids from a sick person are added. The inference being that if the person's sick and if there's a virus in there, if you add the fluids from a sick person, cells are going to break down because of the virus. So what happens? Some of the cells break down in the soup when they add fluids from a sick person, and they conclude we have isolated a virus. That's what they mean by isolation. This is hugely problematic from a logical standpoint. And the logical fallacy is worth mentioning because it comes up all over the place and it's relevant to GNM. It's called affirming the consequent. The formal version is if P, then Q, Q, therefore P. A simpler example is if it rains, the grass outside will be wet. I see that the grass outside is wet, therefore it rained. Whoa, that second sentence is a problem because you've left out every other possible cause of why the grass could be wet. Maybe there was dew. Maybe there were children running by who poured water on the grass. Maybe there were interdimensional beings that popped into our dimension and poured water and then popped out. Like, we could come up with so many different reasons of why the grass was wet. Same thing goes for this study or for why people get sick in general. Modern medicine and modern thinking likes to latch onto one cause and then build everything around that model. So what do they do with this study? They say, we take fluids from a sick person. The person had symptoms. There must have been a virus in there. Look, cells broke down when we added it to the cell culture soup. There must have been a virus. What are some of the other explanations? Well, if you're taking fluids from a person who's sick and you're not purifying the sample fully, so it's not like they're taking just the virus out of the person. There's other stuff in a person that's sick, and who knows what's in there. Maybe there's other stuff in a person who's sick that if you add it to the cell culture, that could cause cells to break down. That's something that would need to be explored. That could have nothing to do with a virus. There could be no virus in there, and the fluids from the person that's sick could cause cells to break down. Another possibility is what if something in the cell culture soup itself would cause cells to break down even if you didn't add a virus to it? There's antibiotics, there's monkey kidney cells, there's a whole litany of things in there. And what's crazy about these studies is that the proper controls are not done. Now, let me preface this by saying that you actually couldn't ever have a proper control without having an independent variable. An independent variable is the thing that you add to a study to see what the effect is. So if you wanted to do the study properly, you would want to have just the virus by itself as the independent variable. And the control would be to look at what happens without adding the independent variable. But if you don't have an independent variable, a true one, where it's just the virus, then you actually could never create a proper control. So you're in big trouble. But what people call a quote unquote control would be to say, let's just look at what happens in the cell culture soup if we don't even add an alleged virus. And not many of these studies have been done, which is pretty amazing. Stefan Lanka ran some studies where he would add yeast RNA to the cell culture soup, and he saw that cells broke down. So you'd want to have many, many replications of this. And there are some studies in the works. Some that are getting a lot of attention now are being led by Jamie Andrews in the UK, and he's been speaking about this on Alex Zek's podcast and other places too. But there will need to be a lot more of these studies of, let's just look at a cell culture. Let's vary the amount of antibiotics. Let's vary what's in there and see if things break down without it. So we've got big problems here because the whole basis of virology is on this sort of study to say we've isolated the virus. And then everything else downstream comes from the belief that the virus has been isolated. That includes any diagnostic test, anything you see under an electron microscope, and anything else you could think of that you would call quote unquote viral. It all ties back to the assumptions made in the study, and that's a big problem. And I want to just mention one thing before pausing, because your audience members might be saying, well, I've seen pictures of the virus, of SARS-CoV-2. It's got all the spikes and whatever. And we've seen all these things under an electron microscope. And it's true, they have seen things. But if you don't have a purified sample, how do you know what you're looking at?
Yeah.
You don't know. And it's possible that if a person is sick with the same symptoms, maybe those things would show up all the time in a cell culture soup. There are lots of other reasons to explain it other than a virus. And I should add that electron microscope technology is problematic for this whole thing because you have to basically distort the sample and tamper with it. So you're taking the results of a cell culture, then you end up slicing it up, you end up changing the temperature, you're shooting it with electron beams theoretically, and you end up looking with a static image. That's critical, static. A virus is supposed to be moving around and getting inside the host cell and causing disease and doing all these things. You're seeing a still picture of something that's dead, whereas a virus is supposed to be moving around and living. So even you're making all kinds of inferences here. I mean, if we wanted to be really specific about this, and I'm sure people will say, well, Mark, you're asking for too much. I don't think so, because a virus is supposed to be this intracellular parasite that goes from one person to the next. Why can't we see that to prove that it's real? Show direct evidence of a person within a person's body, the virus doing all these things, and then going from one person to another and causing disease. We don't see that at all. We see these inferences of, let's take the product of a cell culture and look at it under an electron microscope, or maybe look at fluids from a sick person and look at it, and then make a whole bunch of inferences rather than actually have direct evidence.
So something else that I think might be helpful for our audience to hear a little bit about and to touch on is the subject of testing. Like when they test for viruses, let's just say the PCR tests that were used, in the COVID era, what are they testing for? What makes certain tests come out as positive, right? Especially if that person is symptomatic. I know obviously there were people that were not symptomatic that were getting positive results too. But when someone goes and gets tested and they're like, oh yeah, you have COVID, and then they get feeling better, and a few weeks later, let's say that person has a negative test. What's going on with that, and how do they even construct the test? Like, what are they testing for?
Yeah, it's a great question. That they're testing for things that are supposed to be proxies for a virus. That's basically it, because we know there's never been a purified sample, and all these organizations like the CDC and otherwise and the Freedom of Information requests have verified this. We never have a sample of just a virus by itself. So anything that is downstream from that is going to be an inference. So one might ask about like genetic sequencing. So the PCR test is ultimately a genetic type test. So theoretically, if you knew what the virus's genome was, then you could look for that in people and say, then that we have now found that you have this virus. The problem is that if you never had a purified sample of just the virus, if you end up with a soup of other sorts of material in there, then whatever you're looking for in terms of a genome could never be tied to just a virus. It's going to be picking up potentially other things too. And then separately, there are all sorts of questions about whether or not a full genome has ever been established for any organism, or whether they're still missing pieces, in which case we'd be running other inferences, and whether nucleotides have been isolated. There was actually a recent podcast, Dr. Tom Cowan has been looking into this. He was on the Way Forward podcast with Alec, and he also did a webinar on his channel asking questions. So I'm not saying I know for sure, but there are similar questions that we see in virology with even DNA. But in any event, let's assume that we could have a full viral genome. You would want to have a sample of just the virus by itself. Otherwise, as Dr. Mark Bailey says, you end up with sequences of unknown provenance, meaning their origin is unknown. So this is problematic for any test. So a PCR test is testing for genetic material, but we don't even know what the genetic material of a virus is, or if it's material from other things. So I think the answer to the question is we don't really know what a PCR test is picking up, and we don't know exactly what it's doing. And even Kerry Mullis, who invented the PCR test, he won a Nobel Prize. He said with the PCR test, you can find about anything. And I quote him in the book saying this because some of these tests have to do with like the amount of cycles that they're looking for. And it gets very complex. And this is another reason for my skepticism is the field of genomics is sort of like a black box. Like the computer technology they're using to like basically put together different sequences to say that they have a whole genome or that they detected something. There just seem to be many ways that you could make inferences or that it could go wrong because you're relying on different technologies and mathematical models. And that's why some of these tests are not always reliable. You could get someone who's sick with symptoms who doesn't always have a positive test. So you get these false positives or false negatives sometimes. And it is possible, I'm just going to give a theory also, that if someone is sick with similar symptoms, that maybe their body's producing similar things, and therefore a test could pick up those similar things, and therefore the test is correct more often than not, meaning correlated with symptoms. So there's a principle here also, which is that it's not necessary to come up with a replacement theory when debunking one. And that's really important because we could show how the theory of the virus as defined as this intracellular parasite does not stand up to the scientific method, but it doesn't require us to then explain everything else in the universe because we've debunked this one thing. The way I talk about this in my book on cosmology, I think it's a useful analogy here. It would be like the three of us taking a multiple choice test in school, and the teacher says the three of you need to come up with the same answer and submit it for your for this test. And you two go off and you say, look, there's options A, B, C, and D, and we have to pick one of those. You come to me and say, we really think it's option A. We need to circle that and submit it to the teacher. And I say, well, I've looked at option A all week, and I can show you all these reasons why it's not option A. And you both say to me, like, Mark, you make some good points here. We still think it's A, but you make some really good points. Tell me, is it B, C, or D if it's not A? And I say, Abigail and Ashley, I really don't know. I haven't had enough time to look at B, C, or D.
Yeah.
I just know it's not A. And then you two say to me, well, then we're going to submit A, Mark. It must be A. That's what happens all over the place. And virology is a really good example of that, or even COVID, SARS-CoV-2. People say, well, you've shown that SARS-CoV-2 doesn't exist. Tell me exactly why everyone got sick with these symptoms. It's actually not required to come up with an alternative to explain why something is not true. And it's the same thing with all this stuff. These are great questions with PCR and diagnostics. Why is it positive sometimes? I don't have a full answer for this. And I've heard many doctors say we don't have an exact replacement answer to explain all this stuff, but we know the virology theory that there are holes in it and they're not following the scientific method.
Yeah.
So that's excellent. I'm going to throw out an example here and you tell me if this doesn't make any sense at all and if this is like way off. But say if I'm creating a cocktail, okay, and I'm putting in different ingredients and it's my little custom cocktail and I call it Great Berry Dreams. Okay, I give this name to this cocktail and it's a combination of different things, but I decide to name it as one thing. Like, okay, when all these things are put together, this is what I'm going to give the label to it. You know, I'm going to call it this. So someone else could be like, well, Great Berry Dreams, that doesn't exist. Like that, you know, that's not real, right? And they're right in the sense that maybe for them, I don't mean for them it's not real, but like based on how I have decided to name the arrangement of these things in this cocktail, this is what I decide to call it. So is this a little bit like what they're doing in virology where they're like, oh, this, I don't know, evidence of what we think is a virus, like what we're looking at, that's like this, I don't know, evidence of a virus, like that thing, is it even important? Like, is it a virus or not? Because they're just, whatever they're seeing, they're calling it a virus. When I guess what we're saying is that more what we're disagreeing with is that, okay, if they want to call that a virus, they can call it a virus, but we're disagreeing that viruses are actually the cause of disease. Does that, I don't know, I feel like that is a little convoluted, but
Yeah, let me try to summarize here because I've heard
okay.
some people push back and get very angry at what I'm doing and say, But who cares if it's a virus? People are getting sick. It's like something gets complicated in what we're actually discussing here. There's a claim that's been put forth by the medical community. It's a bold claim that has led to people losing their jobs and people getting shut down and censored. The claim is that there is this intracellular parasite organism that gets into a host cell and causes disease and goes from person to person. We're asking, has that particle been shown to exist, and does it actually cause disease by itself? Instead, we do get this sort of cocktail, Abigail, and I don't know if this is exactly what you're describing, when we take fluids from a sick person, and it's a cocktail of things in the person's body. But until the cocktail has been reduced to just the one ingredient that we're looking for, which is the virus, not the quote unquote antibodies that they claim is a result of the virus, which you would need to have a virus in order to show that anything came from it, whether it's an antigen or an antibody, you need to have a virus first or a spike protein to show that those things are linked to the virus, which has never been established. We want to see all the ingredients in the cocktail, basically, but actually we just want one ingredient in the cocktail of what comes from the person, which is the virus. And one other point that I just want to raise here is that, I just remember how I used to think about this. Like, is this a psyop? Is this to distract us? Because it just seemed to pop up around COVID that everyone was asking the question. But what I didn't realize until I started researching is that this was a big issue around HIV/AIDS.
Yeah.
Too. And it was a little bit smaller because HIV/AIDS wasn't the same level that COVID was, where the whole world was being shut down. But HIV/AIDS was a big deal. And there was a group of people who said this retrovirus that's allegedly causing symptoms, there were symptoms. The question was it caused by a retrovirus? Some people said no, it wasn't caused by that retrovirus, and there were many other reasons. And then some people said that retrovirus has never been established. And there's an organization called the Perth Group at theperthgroup.com. They've written a paper called HIV, a virus like no other, in case your audience is interested. Very technical stuff. But the point is, if you look at the history, 1954 was the Enders and Peebles study, which was right after the 1953 Watson and Crick paper. And this was also around the time that bacteriophages were established, and actually there was a Nobel Prize awarded for it. I believe it was the next decade that the Nobel Prize was awarded, but this was being researched around the time. And the bacteriophage is what's believed to be a virus-like particle for bacteria. So the reason that the thinking was, oh, there's a virus that infects bacteria. Maybe there are other viruses also that are DNA linked. The problem is that some people say the bacteriophage particle is actually a byproduct of a bacteria. It's not actually a virus. It's not infecting. But the point is, you had this confluence of events around the 1950s. And then, so this was a new concept, that there was this virus particle specifically that is infecting host cells and everything. And then the early 80s was HIV/AIDS. So you could see how this is a relatively recent invention, basically, the virus thing. And then COVID was the next big one. So it actually, to me, makes sense that we could get it this wrong because it's so new and hasn't really been, people haven't tried to poke holes in it. And it gets to, I think you make an important point, Abigail, of like really honing in on what is the claim that's being made and being questioned. There's a claim, show me this particle exists by itself and causes disease. That's what Tom Cowan says. He's asked people all over the world, how do you know that a virus exists and causes disease? And I've started to ask people this. Doctors, oh, well, a positive PCR test. Oh, a bunch of people got sick in the same place with similar symptoms, and therefore they must have caught something. That ignores all the other ways that people might get sick, including GNM explanations. But that's really what we're dealing with in many ways is psychological, is getting people to wrap their heads around what is the claim that's being put forth, what's the history of this claim, and why do people believe it's true, and why should we question it in terms of the scientific method?
Yeah. Well, Abigail beat me to an analogy. I was going to make a soup analogy with ingredients, so you beat me. Yeah, so I wanted to go back a little bit and ask a question, for our listeners. So, we're talking about that a virus has not been able to be isolated. And so in your research and knowing what we know from medicine and all of these studies and the electron microscope, we know that bacteria have been isolated. So can you talk about like the difference between, these scientists trying to isolate a virus versus them isolating bacteria, and and maybe the methods were different, or just the fact that they were able to do it with bacteria?
Yeah, it's a great question. So bacteria are much bigger. That's really what it comes down to. Viruses are supposed to be so small that you need to use this electron microscopy technology, which ends up distorting the sample in order to see
Okay.
it. Whereas bacteria are much bigger, they've been established for a long time. But then the question becomes, how do we know that bacteria are actually causing disease
Yeah.
versus acting as some kind of a helpful mechanism of the body? That's a really important question that I still get a lot of pushback on. And to me, I think it should be an easy to prove thing again. And there are steps that you would use to show this logically. They're known as Koch's postulates. And the WHO has said very recently that this is the gold standard, basically, and I quote them in my book, because some people would say Koch's postulates are outdated. It's true that there was a revision for viruses known as Rivers' postulates, which are very similar. But basically, Koch's postulates are the first step is that you'd want to show that these bacteria that are causing disease, they appear in sick individuals, but not healthy ones.
Mm,
That
yeah.
right there causes lots of problems because you could go through bacteria after bacteria where they're in healthy people too, and then something happens and they appear in large quantities in people who are sick, which indicates there must be some other factor that's causing them to appear in large quantities. But ignoring that, how would you prove that something causes illness, or at least try to using logic? You'd want to have a sample of just that thing, of just the bacteria, and you'd want to see it in the sick organism, and then you'd want to put it in a healthy organism and then re-isolate it in the healthy organism and show that that also caused illness in the
Mm-hmm.
organism that was healthy, that they got sick after this thing was introduced, and then you re-isolate it after you see it in this now sick organism that used to be healthy. Those basic steps. And what seems to be the case is that that's not being done. All those things are not being followed, including the first step, which is that the bacteria only show up in sick but not healthy organisms. And there are some freedom of information requests that have been submitted on this topic, and I'm sure more will occur with Christine Massey and her colleagues saying, show me that this is a disease-causing bacterium using Koch's postulates. Show me the fulfillment of Koch's postulates. And they come back and say, we haven't done that. So, I mean, with all this stuff, I'm open to new papers emerging, but it's just shocking to me as just a normal citizen that there wouldn't be thousands of them, like thousands of them for every alleged bacterial illness and every alleged viral illness, not just like one paper where you can debate the merits of it. Thousands and thousands, binders and binders of papers. So that raises a lot of questions for me in terms of what are bacteria, and it aligns with GNM. And one of the reasons I was so open to what I heard about GNM is that wow, it actually aligns with these microbes might be helpful. And if what we call a quote unquote infection, yes, there are many bacteria appearing in large quantities. And yes, bacteria might emit endotoxins, but the bacteria aren't the root cause in those cases because the bacteria are serving a function in the body because there's an underlying toxicity, which in GNM starts with the mind that ultimately corresponds to organs. But the bacteria are therefore helpful. So that is a big shift for people. And it doesn't also mean that an antibiotic wouldn't help alleviate symptoms in the short term. It might, and maybe you need to do that because there's so much toxicity and there's so many bacteria that you need to do something in the short term. But that still doesn't prove that the bacteria are they're the root cause of the illness.
So going back to how, because I actually don't know the specifics on this, but how were bacteria discovered? Like, I guess what I'm trying to get at is when they went to, did they set out to like find, like, did they have this theory? Oh, there must be this thing called called bacteria. There must be bacteria, right? And then they set out to try to go find it and like prove it, which isn't that kind of what they're doing with viruses? They're theorizing, oh, there must be this thing called a virus, and we're now going to set about to find it. Was it the same with bacteria, or did they, in the research they were doing, they find this thing, this particle, and they're like, oh, we're going to call this, we're going to call these particles bacteria, right? And then I guess.
Yeah, no, it's a good question. I'm actually not sure which came first, whether it was just the technology existed to see microscopic things with the lesser advanced technology, and they saw these things repeatedly in people who were sick, or whether they were actually looking for it to see if there was a physical cause. I'm not sure which came first, and there was probably a little bit of both. It's my guess. But I think what you're getting at is this desire to find a physical cause of illness, something that's transmissible. It's a very reductionistic perspective.
Yeah.
That there must be something physical that goes from person to person, and therefore you caught it. Like, we use that term very often. I went to a party, a bunch of us caught something. Embedded in that word caught is the belief in this physical thing that went from person to person. So I do think that that philosophy, which comes from scientific materialism, and we'll talk about that a bit more in the next episode, that everything has a material cause. And therefore, if you can find the material cause, you can eradicate it. It's very simplistic rather than believing that maybe there's something else going on. So I do think that is what happened with viruses too, is they said, well, we know they're bacteria. We know they're associated with illness. We can find them in large quantities when people are sick. Maybe there's this even smaller thing that's transmissible, that's causing illness. So we're going to try to find that.
Yeah. Yeah, well, and we can get more into this in the next episode, but I just wanted to touch on, the fact that germ theory, it makes all of these companies a lot of money. And when we're outsourcing the reason why we're sick and we need to outsource, what do we do about it? Oh, I need to take this pill. I need to do this surgery. I need to do XYZ in order to fix my body or my symptoms or a dis-ease. That is making all of these companies in the medical system so much money. And when we are essentially giving our power away and we don't realize, what the root cause is of why we have the symptoms we do, and we're looking to all these outside things, it makes perfect sense that dismantling this whole paradigm of medicine in general, like, people don't want us to do that because it's making all these companies so much money. And not even just like changing your mind, but it's like, again, following the money, you're going to see where they've put money into research, and they want to research and keep studying what is already making them money. So if it's this outside thing, if it's the virus, if it's a bacteria, if it's this outside particle that we can, quote, prove causes illness, which they haven't been able to do. But if we can make people believe that, then we will come up with the solution, right? Whether it's a antibiotic or a vaccine or anything else. I find it so fascinating, and it's a big reason why I left the medicine system. I used to be a registered nurse, and after learning GNM, I left like 6 months after, because I was like, ooh, this does not work, at all. And yeah, but I wondered, what resistance you came up with when you were researching, or stuff for your book, and even asking doctors the questions you were asking them. Like, what resistance did you come up against with the money piece?
Yeah, I think in my private conversations, I didn't get as much resistance on the money piece, but I do think this general movement to question germ theory is not going to be endorsed by mainstream interests who are funded by pharmaceuticals. Because the implication here, and this is why it leads to GNM, is that if you believe in germ theory, then there's got to be an external savior to help you eradicate this invisible enemy. Therefore, you offload your personal responsibility to a third party or something else. Whereas if the roots of disease are ultimately psychic or related to our consciousness, and that flows into our body, and yes, there are material things that contribute to all of it too, then it puts the onus much more on the individual.
Yeah.
And that doesn't make as much money for large industries. I mean, think about virology is a field that is based on a particle that has not been established using the scientific method. How many, number one, jobs and financial interests, but also people's own egos and their careers that have been spent on this basis. But when I've had one-on-one conversations, primarily with doctors, I think virologists are much more defensive because they're actually doing stuff with the test tubes and everything. And to think that maybe they're not doing proper controls, or maybe it's not exactly what they think it is, that they've been doing, like, because I think many people are perpetrating this falsehood innocently. There might be some there obviously some who know there's a problem, but I think others are just following the methodology. Oh, this is the gold standard. Like, what are you going to do if you're a PhD student? You're going to question the gold standard that the whole field is based on and say maybe there's no virus. And that's why Stefan Lanka's work is so remarkable for a virologist to switch,
Question.
but to question it. And now he's been treated terribly as a result of that. And even I should mention before, two things. Doctors, when I talk to them, I've found they're a little bit less resistant because they realize they haven't looked at the virology studies, and they say, actually, yeah, viruses are often just a cop out. Like if I don't understand why someone's sick, I just say, well, it must be viral, as if that's some kind of
Mm,
like profound statement that they understand there's a lot we don't know. And in med school, they were just trying to get through and they listened
yep.
to X, Y, and Z. But I wanted to mention Stefan Lanka, who has been, um, he's taken a lot of heat, and I think he's very brave, but he put out a challenge for the alleged measles virus because he was very upset about the measles vaccine. He offered $100,000 to someone who could submit papers showing that the measles virus exists. And someone submitted 6 papers, and Stefan Lanka said, no, this you didn't prove the measles
This
virus
isn't it.
exists.
Yeah.
This is not it. So he was taken to court. And the court initially ruled against Stefan Lanka, but he appealed it. And this was in Germany, where the appellate court has more technically minded judges, and they ruled in his favor. He said that the papers that were submitted did not appropriately prove that the measles virus exists using the scientific method. So that was a pretty big stamp of approval for me because, again, shouldn't this be a slam dunk? Shouldn't they submit papers and show it? And in the book, in my book, An End to Upside Down Medicine, I go through the 6 papers that were submitted, and Dr. Sam Bailey in New Zealand goes through like why each one is not proving that it was a measles virus that was detected. So these are really like these things, as just an independent researcher, they shouldn't be happening. And yet they
Yeah.
are. And where is just the mountain of evidence to contradict all this stuff? It's never there. And one more point I'll make before I pause is that I sometimes play around with artificial intelligence, which has access to so much data, like ChatGPT, so much stuff. And they're supposed to follow logic. And when I present it with this information, I say, here in the Enders and People study in 1954, they actually say that there might be unknown factors that cause the cells to break down in the soup, that even they, in their own words, they say we didn't prove that it was a virus. There might be something else. So I present the AI with these things, and AI is forced to say, you're right. Following the scientific method, we don't have what you would need to have science, meaning a true independent variable and proper controls. And therefore, dare I say it, if you're not following the scientific method, you are engaging in pseudoscience. And that is devastating for an entire industry and field that makes a lot of money,
Mm-hmm.
that it's based on pseudoscience and we were just being told to trust the science. It's highly hypocritical.
Yikes. Yeah.
First of all, I love your Instagram post where you post your conversations with ChatGPT. I think that that's fantastic. So guys, make sure that you're following Mark on Instagram. Very, very entertaining and informative. So I remember reading in your book about a certain study, and I found this to be absolutely mind-blowing, where there was, I guess, a team of researchers that they were trying to um, conduct a study with natural methods of supposed contagion, right?
Mm.
Like natural methods of transmission. So, as opposed to previous studies, I'm assuming where they had, taken fluids out of a sick person and then the soup and all this, they were trying to say, like, look, let's just try to have this transmitted in a natural way, because isn't that what this theory of contagion is really all about, right? So they put people, you can describe it better than I can, but I know you know what I'm talking about.
Yes. Yeah. So I want to give context here. I'm glad you brought this up, Abigail, which is we have been previously just been talking about whether a virus even exists at all. But then there's another part of the claim, which is that the virus exists and that it can be transmitted from person to person. But I have just been stunned in the same way I've been stunned about the lack of isolation of a virus, stunned at the lack of demonstration of contagion, which even if you have an isolated virus, let's just say you take a bunch of sick people and put them in a room with healthy people. Do the healthy people get sick? What are the studies done on that? And the study you're referring to, Abigail, was done by Milton Rosenau. This was around the time of the Spanish flu. So like roughly 100 years ago, where like, remember, this was supposed to be like the deadliest virus ever.
Mm-hmm.
And they tried everything to transmit this alleged virus, and they were unable to do so. And they even had patients cough directly into the faces
Yep.
of healthy volunteers many times. They injected fluids into people who were healthy, and these were fluids from people who were supposed to be sick with symptoms, and they were never able to make the healthy people sick. And this leads to a broader category of studies of basically failed contagion. Um, and there's a new book out. I believe it's called Can You Catch a Cold? It's written by Daniel Roitas, where he goes through all the alleged contagion studies trying to demonstrate the the contagion of a cold. And over again, the studies failed to do so. And how is contagion alleged to be demonstrated then? Because clearly something is making doctors believe that some one particle goes from one to another and makes the healthy person sick. And it's again using sloppy science. They are typically injecting an unpurified sample, and in the case of viruses, it's by definition unpurified because they've never given us a sample of just a virus. So it might be a cell culture, which has a bunch of other stuff in it, not just an alleged virus, and they're injecting it in animals in very unnatural ways. So think about it, like with COVID, we were told that at first that it could exist on surfaces and just by touching something maybe you'd get it, and then it
Yeah.
was airborne. You could just be in the same room, very delicate things, and that's how you get it. That's not what they're doing in these contagion studies. They're injecting directly into an animal's body this cell culture soup or other unpurified sample. And they'll do it by injecting it in the brain of an animal or in the trachea. And sometimes, believe it or not, the animals start to exhibit symptoms, sometimes not even exactly the symptoms that they're looking for, but sometimes just generally bad symptoms. And they say, look, we just proved contagion. Shouldn't you run controls? I mean, ethics aside, there's big ethical issues with like injecting an animal with fluids. But what if you injected water into an animal's brain or trachea? Would that cause symptoms? Or what if you injected a cell culture soup without an alleged virus in the animals? So they're not running the studies properly. It's extremely sloppy science over and over again to allege to say, oh, we just showed it's contagious. So when I have conversations with doctors, it's the same kind of conversation that I had around the isolation of the virus. If I say, how do you know the virus exists? I don't know, like this PCR test. They're not talking about the isolation methods. And if you say, how do you know contagion was established? Well, they did those studies a long time ago. But oh yeah, I actually haven't gone back to look at how those studies were conducted. I haven't looked at the methodologies that were used. So we end up, what I'm finding is jumping to conclusions based on proclamations by a small number of scientists. And we see it very clearly with SARS-CoV-2 because that has been looked at under a microscope. There's a great paper that's available free online. It's called A Farewell to Virology by Dr. Mark Bailey. And he goes through some of this with regard to SARS-CoV-2, like what was the original paper done on the genome, for example? How did they find the SARS-CoV-2 genome and everything else? And you just see the sloppiness of the science. But then what happens is a paper comes out and the title of the paper says isolation of SARS-CoV-2 or something else that proclaims something. And everyone says, oh, it's been isolated. And what's happening, what's not happening is people aren't going into the details. And I can understand why, because the terminology is very hard to understand. It's super complex. And it's taken many of these brave doctors to break it down for us and to expose each word of the document of these fundamental papers and say, look, this is where they're claiming X, Y, and Z. And then now what's happening is people are starting to ask questions.
Wow. So just to while we're on this topic, to follow up, the scientists, the researchers that are, I would assume that at least some of them would see how these studies are not exactly being conducted in a great way, or the studies that were conducted where they're like coughing each other's faces and no one's getting sick. There are very few people. Are they not questioning that? Like, I guess in the scientific research community, the people that, I'm not talking about doctors, but just doctors that this is not their specialty, but the people who this is actually their specialty, are they trying, like, are they being intentionally misleading? Are they being paid off? Are they just unintelligent? Are they blindly following authority? Like, what's going on here?
It's a great question. It's probably a little bit of everything you just said. I don't know what the breakdown is. Like if you presented me with any one virologist, how can we know what their actual intent is? Because I do think there are some people who just firmly believe what they've been taught and they have a PhD and they've been doing this for years and they see cells break down in that cell culture soup. The cells broke down. It's really hard sometimes to reverse engineer and ask questions. So I think there is a lot of ego that's wrapped up into it. And then you might have people who are afraid to speak up, understandably, to say, ooh, I'm not sure about this methodology, or maybe we should use a different methodology. And the powers that be above are probably saying, no, you got to do it this way. And then there's the other element of scientific journals and which papers get promoted. So it could be that maybe there are some who ask questions, but you have scientific journals who are only wanting to publish the stuff that validates everything because they're part of the whole medical industrial complex. And then what do you know? Certain papers, those are the ones that get certain scientists very famous. And those are the ones that use certain methodologies. So I think there's a lot of self-fulfilling stuff in this too.
Well, and two, even when I worked as a nurse, we we had to distribute the flu vaccines, every fall. And I actually got in trouble because my numbers were less than like the average of the other nurses on our floor, because they track everything in the hospital. So basically my manager called me into her office and was like, why are you not giving people the flu vaccine? I was like, what do you mean? And she's like, oh, well, your adherence numbers are like not up to up to snuff. And I was like, okay. And I was like, well, it's optional, right? And and she was like, well, yeah, but we want to like highly encourage our patients to get the flu vaccine. And all I said again was, I said, it's optional, right? And I said, I'm letting my patients decide, and I'm giving them the whole form, not because we were actually taught to fold it in half. So the side effects were inside the folded part, and give it to our patients that way. So they're only seeing the benefits and like why you should get this flu vaccine. And so I wasn't necessarily encouraging my patients not to get it. I was just like, first of all, it's your choice. And like, here's the information. You make a decision on if you want to get it. And so like, naturally, because I'm not being coercive, not everyone said yes. And then I get reprimanded, or I got in trouble. Then this happened like with like a couple times within like the flu season, where again they were probably like watching my numbers more. And I got in trouble for it multiple times, and it wasn't because I was telling people not to get it, but simply because I was giving my patients a choice instead of saying you have to get this, or else. And so even as just a normal registered nurse on a hospital floor, I wasn't like doing studies on these bigger things or trying to disprove things, but just even just me trying to give my patients some semblance of, here, you can make your own decision, like then I'm getting in trouble for that. So I can't even imagine like the pressure on these researchers or on these medical doctors, where they're seeing things differently than what they've been taught, but yet, right, their livelihood is threatened. Their reputation, their ability to, even work in a different field if something, catastrophic were to happen to their reputation, it's like that follows you. And so, yeah, even from my own experience, I can't imagine how some of those people must be feeling, even if they do see the validity in questioning things. Like, what can they do? Yeah.
Yeah, and when people's livelihoods are dependent on this stuff, sometimes they might see something and say, well, I need to feed my kids. I'm not going to risk my whole career. So you need a perfect storm, really, of someone who is knowledgeable enough and then willing to be lambasted and lose a paycheck over this. And so there have been very few, but I think as this information has gotten out, I mean, I think about where I was in 2020 versus where I am today. I wasn't willing to look at this and really speak out, but it took some time of seeing the validation of more and more doctors, and to be able to put together the end of COVID at the end of COVID.com, to see all these professionals seeing the same thing and giving data points. And now we even have people like Michael Yeadon, who was a former VP at Pfizer, speaking like this, questioning viruses, and some other prominent figures that are at least asking the questions, or they're not being as antagonistic to it. So there is momentum, but this is really sort of like GNM. It's like a very small corner of the medical freedom community relative to other things. There's a lot of people asking questions about vaccines, but how dare you question viruses? There are a lot of anti-vaxxers who would be upset at this conversation, for example.
Yeah.
And who would be upset about GNM also.
Yeah.
So it's like, this is really difficult stuff given where we are today. But in the same way that 4 years ago, 10 years ago, the anti-vax stuff was total heresy. And now, just yesterday, RFK Jr. was nominated to be the head of HHS. There's progression in terms of what is able to be questioned. And that makes me optimistic about understanding the roots of disease, viruses, and GNM also.
Yeah, absolutely. I think something that I'm still, my brain gets like stuck on certain things. I'm still just really playing with this question on like, who is defining what a virus is? Because I know that you said it was initially defined as a toxic slime, right? So how did it go from that to getting mixed in with this concept of contagion? I guess that's what I'm really trying to understand is like, how did that even come about? Are they, and I guess what's the motivation? Is it
Yeah.
just we need to find something external, or sorry, something transmissible that we can blame disease on? Like, I guess where did the idea of contagion come from?
I think it comes from, this is my guess, two things. One is materialism, the idea that there's a material cause to things. And then two, there's a there's a way to control people. If there's a material cause like
Hmm.
this invisible enemy that you need to offload your authority to prevent, because there's
Yep.
nothing you can do. It's sort of like the genetic determinism too. Oh, there's nothing you can do. It's just your genetics. You better
Yeah.
go to this doctor. Oh, there's nothing you can do. There's this tyrant microscopic thing that's going to come get you. There's a product that can save you, and that's the only way you can be saved. So I think that it's twofold, and they go together because I think materialism is part of a control mechanism too, versus understanding the broader reality, things beyond what the eyes can see and beyond what's material. But in terms of this notion of a virus changing definitions, when I heard Stefan Lanka discuss the 1953 Watson and Crick paper, that was a real light bulb for me, because there was this notion of a virus, or even with Pasteur, a transmissible agent that we just can't see. But now with DNA, there was a mechanism for it of, wow, it's genetic. If you get inside the cell, then you can do all this stuff with genes and all these complicated things, and it's a genetic enemy. So I think there was a lot of incentive to come up with that, and then genetics gave a new framework, and the bacteriophage also, like I mentioned, because they said, oh well, there's a virus for a bacteria, even though some people would dispute that. Now we can come up with this really scary thing to explain illness, and now we've got it. Again, I don't think it's everyone that was probably so nefarious with it, but it does.
Mm.
I think there might have been some that
Yeah.
that wanted to push the narrative. I do think a lot of these narratives are
Mm.
pushed by innocent people, though.
Yeah, that makes sense. So one more thing that you mentioned in your book before we start to wrap up with this part one episode, you mentioned the proof of burden, right? And when we talk about like proving that a virus exists, can you talk about how this proof of burden really is not on the viruses don't exist camp?
Yeah, it's a great point, because what's happened is a claim is put forward. Some call it a positive claim. And the positive claim is that there exists a particle that's microscopic called a virus that gets inside of a host cell and replicates and causes disease and goes from person to person. That's a claim. It's a scientific hypothesis that needs to be shown. And what happens often is that this claim is believed as law, basically. So when you say there is no virus, they say the burden of proof is on you because it's already been established. No, anytime a claim is put forward, that's where the burden of proof lies. So, and then the next part is, well, if you can't tell me, if you say it's not a virus, then you need to tell me what it is that's causing people to get sick. That's another logical fallacy to say that you need a replacement. So again, this conversation here is very specific. A claim has been put forward to all of us, and it's a very important claim because a lot of fear is built around it, and the world is being shut down on the basis of it. And there are medications that people are being coerced to take and inject in their arms as a result of this thing. It should be, I don't understand why people get so upset by it. Like, just show the evidence very clearly. Show an isolated, purified sample. And what I continue to see everywhere is excuses that are being made for this.
Yeah.
And then you have these government agencies with the freedom of information requests explicitly saying we don't have evidence. So to me, that's enough to make this a legitimate issue that needs to be explored.
Yeah. Yeah. Well, as we wrap up this episode, I just wanted to say thank you so much to our listeners and to Mark for this part one of our interview with him. So stay tuned for next week's episode with Mark on if it's not germs, then what is it? And we'll see you next time.
Bye, guys. We hope you enjoyed today's conversation. We appreciate you being here.
If you would like some additional support in navigating your health symptoms, we both provide coaching services from a German New Medicine perspective.
Our Instagram handles and personal websites are linked in the podcast description.